Welcome to our video on a cornerstone technology in modern biotechnology, producing recombinant protein with the Baculovirus-Insect cell system, or BEVS. Whether you are new to this field or seeking deeper understanding, this session will explain why BEVS is essential for producing complex proteins. Our story begins in agriculture. Baculoviruses were first studied for pest control before their potential for protein production emerged. In the 1980s, researchers, including Max Summers and Gail Smith, made a breakthrough by replacing the polyhedron gene with foreign genes. This simple idea transformed baculovirus into a versatile vector for high-level recombinant expression. Continuous refinements followed. Modular systems like back-to-back and flashback streamlined cloning and virus generation. These improvements made BEVS a gold standard for producing complex, correctly folded proteins. Today, BEVS is widely used across research and industry for its yield and flexibility. Next, we'll cover the biology behind baculoviruses and how the system works in cells. Let's define baculoviruses and their safety profile before we examine their role as molecular delivery trucks. Baculoviruses are DNA viruses that infect insects. They exist as two virion forms, budded virus and occlusion-derived virus. Crucially, they are classified as biosafety level 1 and pose no known risk to vertebrates, making them safe for lab use. Baculoviruses function as molecular delivery trucks. Large genomes, high DNA capacity, and safe profiles for expression. ACMNPV is the workhorse species, efficient at infecting insect cell cultures and accommodating foreign DNA. They are mammalian systems, adding another layer of safety for recombinant protein workflows. The replication cycle has distinct phases. First, early genes reprogram the host to support viral replication. Later, late genes drive DNA replication and produce structural proteins to build new virions. BEVS exploits very late hyperactivated promoters, generating very high levels of target protein expression. Next, we'll see how baculoviruses are engineered into vectors for recombinant protein production. Using vectors like fastback and systems like back-to-back, foreign genes are inserted into baculovirus DNA to create expression viruses. Engineering converts baculovirus into a protein factory. Insert the gene, generate infectious virus, and amplify for production. Choose your factory wisely. SF9 and SF21 are robust, easy to manage, and widely used. High-5 cells often give higher yields for secreted proteins and are a go-to for some applications. For human-like glycans, glyco-engineered lines such as mimic SF9 can be used depending on your needs. BEVS advantages include eukaryotic fidelity, high productivity, speed, safety, large genome capacity, and cost efficiency. Mechanistically, infected insect cells handle folding and post-translational modifications, which supports functional proteins. Typical workflows peak around 48 to 72 hours post-infection, delivering rapid turnaround compared to some mammalian systems. The stepwise process. First, construct the vector and verify your sequence. Second, create infectious baculovirus and amplify to working stocks. Third, infect production cells, express the protein, and purify for research or industrial use, often within 3 weeks total. In summary, BEVS is a versatile, efficient system for producing complex recombinant proteins. Thank you for joining. Reach out with questions or for more resources on BEVS.